It's always a wonderful meeting, this meeting, and it's really a privilege to be here to be allowed to talk about our programs. So I'm going to talk about Domagrozumab, which is our anti-myostatin antibody currently in a phase 2 study. So this is a monoclonal antibody to the protein local hormone called myostatin, and I'll talk about in a few minutes. So we believe this mechanism has the potential to delay progression in DMD, and we expect that it will do so regardless of Duchenne genotype. The study, 516-1002, as we like to call it, is an ongoing phase 2 study that is actually potentially a pivotal study in the views of the regulators. And it's currently running at 34 sites in 8 countries around the globe. And just at the end, I'll give you a little bit of information about the AAV gene therapy platform that Pfizer has been building and news about the DMD gene therapy program. So myostatin is a small hormone that acts to limit muscle growth in normal physiology, and it works by interfering with protein synthesis and degradation through a complex signaling pathway. It's produced by skeletal muscle and acts on skeletal muscle, and is highly conserved in evolution. So we see this in all species examined, and it has a common function in all species that have been looked at. At a cellular level, it acts to inhibit satellite cell proliferation and myoblast differentiation, and in that way inhibits the expansion of muscle in response to exercise. So the therapeutic idea is that inhibiting this hormone may reduce the inhibition of muscle growth and allow growth and regeneration to occur. So the myostatin was described in the late 1990s, and very rapidly one Catherine Wagner twigged that it may have some influence over the phenotype of Duchenne, and her group ran some experiments crossing the myostatin genetic knockout mouse with the MDX model, and was able to show quite a marked impact on the phenotype in muscle, particularly with a reduction in the degree of fibrosis and fatty replacement that was seen in the muscle. And these data were recapitulated with a therapeutic intervention, that is, injecting an antibody into the mice, into MDX mice, to show that even without the intervention in development in a mature animal, that it was possible to improve the muscle quality in the MDX. So with our antibody, we've shown that in mice it can increase lean body mass, increase the amount of muscle, both in sort of normal strains of mice and in the MDX, and as well as that to increase grip strength in the MDX mouse model. We've also looked in non-human primates to show a dose-dependent increase in lean body mass that is actually sustained beyond a short dosing period. These are in normal cynomolgus monkeys. There's also a significant and dose-dependent increase in quadriceps muscle volume as measured by MRI. So this antibody was taken into human volunteers to evaluate the effects in people, and we were able to show that after just 3 doses, we could measure by MRI a meaningful difference in the ratio of muscle to fat in these volunteers after a couple of months. So that provided us with sufficient confidence that we had an active pharmacological drug on this pathway, and that the pathway was relevant and could perhaps influence the disease phenotype. And we initiated this study to evaluate the safety and tolerability and efficacy of this drug in boys with Duchenne. It's quite a complex study, but we've been enrolling boys in the age range of 6 to 16 who are able to walk and on stable dose of steroid. They receive monthly intravenous infusions that last about 2 hours. And we've been looking for about 105 boys to recruit into this study. Throughout the dosing period, there's a close eye kept on safety with periodic reviews that actually initially had to be satisfied to permit a dose escalation. The primary analysis will occur when all the boys have completed 1 year of treatment, and our primary endpoint is the 4-stair climb, but we're also measuring other functional endpoints as well as MRI and other biomarkers. As you can see from this design, it allows for a placebo control period of 1 year, but those boys receiving placebo then receive the active drug. And within this study, we also get some longer-term efficacy and safety information before boys roll over into, if they wish to, into an open-label extension study that is currently running. This whole process is reviewed on a quarterly basis by an external data monitoring committee, and they look at any safety events that are occurring and will be involved in an upcoming interim analysis. So the great news is, from our point of view, is that we've completed recruitment into this study, thanks to the efforts of the boys and their families and their physicians and their study coordinators and nurses and physiotherapists at these sites. Many people involved in this study. It's a huge thank you from us for helping us to achieve this objective. Okay, so I wanted to say a few words about data quality. So we're very keen to ensure that the efforts and sacrifices that boys and families are making in going into this study is reinforced by a very serious attempt to collect the highest quality data possible. So in preparation for this study, we have a very detailed manual of how to conduct these functional assessments, and have instituted a regime of reliability testing to ensure the quality of these evaluations. So each clinical evaluator at each site is individually trained by one of a cadre of 8 to 10 master physiotherapists extremely experienced in evaluating Duchenne patients, and then is tested for inter-rater reliability and requires to meet a certain standard before they're certified to be able to take part in the study. And for this process, we've had many boys volunteer to be tested and assessed, not as part of the clinical study, but really to help the integrity of the study. Each functional assessments, well, not each one, but at critical time points, the functional assessments are video recorded, and those videos are reviewed by our master physiotherapist to confirm that the tests have been conducted to optimum quality. And if quality isn't optimum, then retraining is offered for those clinical evaluators. So this procedure to ensure high quality is, I think, bearing fruit in that we're seeing really very good correlations between visits. Here I'm comparing the screening visit against the baseline visit for stair climb, and really extremely robust data. I'm going to get geeky for a second just to talk about statistical estimate of variability that is used in the literature and to evaluate some of these functional endpoints. And what's been published around the four-stair climb measure is a figure of around 2.2 seconds, that being the smallest difference that you could reasonably be confident was real. It doesn't tell you about the clinical significance, but from a statistical point of view. And from analyzing our data set within a similar age range to the data that was looked at before, but probably slightly better controlled from the point of view of steroid treatment and functional performance going into the study, suggests now that it's nearer about 0.6 seconds as being the smallest unit of four-stair climb change that could be meaningful. Okay, and that's just the equation that's used to estimate these parameters. We have similar high-quality data for the 6-minute walk test, and you can see that there's a reasonable proportion of this population falling into what we're calling the sweet spot of functional activity, where there's probably greatest sensitivity to change. A couple of other things from this study. We've been looking at cardiac troponins really as an exploratory measure so that we can learn about cardiac biology even in boys in this younger age group. And we were actually quite surprised to see that in boys going into this study, even in the 6 to 10-year-old age range, that about 20% of them have really quite markedly elevated cardiac troponins with normal heart function and no cardiac events associated. But it's an area of biology that we believe is very important so that we can learn how best to intervene when therapeutics are available in the future. So what's next? We have an interim analysis that will be conducted later this year. This is when half the boys have been treated in the study for a year. And it involves a number of statistical tests that will be evaluated by our data monitoring committee when they look at unblinded data. So they'll be able to tell who's been treated and who hasn't, unlike anybody in the company. And they will make a judgment about whether these tests have been satisfied and whether it's reasonable to continue. So we don't want to continue a study if it's clear at this point that there is no benefit. Obviously, we expect and hope that there will be benefit and that the study will continue as planned. But it is possible that this drug isn't doing what we expected, and that we'd have to terminate the study. So that decision will be made by the external data monitoring committee, which includes a patient advocate who incidentally is related to this organization. Okay, if I have just 2 minutes, is that all right?
So we've lost some time, so we might need to compress a little bit.
Okay.
Oh, sure. All right, so just trying to...
was just to give you an update on where Pfizer are with adeno-associated virus gene therapy. So this is an area we believe is ripe in development, with the first treatments of this type being approved for other diseases in Europe. This area has been Very challenging, it's exciting prospects for 40 years, but a lot of setbacks. But it does seem as though the field have learned how to generate viruses that are well tolerated and don't cause unacceptable toxicities. So Pfizer have partnered with several companies to bring therapies into the clinic. First of all, with Spark Therapeutics in hemophilia B. And that phase 1 trial has performed very well, and there's some very exciting data around the generation of factor 9 levels in the circulation for sustained periods of time. And this project now has breakthrough status with the FDA and is going on to pivotal evaluation. We've recently partnered with a company called Sangamo, who have another hemophilia A, another adenovirus for that disease, and those phase 1 studies are just about to start. And in August last year, Pfizer purchased a company called Bamboo that was set up, founded by Jude Samulski. And one of the key stars in the portfolio at Bamboo was an adeno-associated virus construct that had been designed for the treatment of Duchenne muscular dystrophy. So this project is now moving forward within the Pfizer umbrella, and we hope to be able to make some exciting announcements about that program and studies within a few months. You will be able to hear a bit more information about those studies and that drug in the gene therapy session this afternoon from Catherine Beaverson. So with that, I'll open for questions.
Actual questions at the end.
Okay, thanks.